Hey everyone, it's Mr. Wolfe here, and in this video I'm going to be going over the first part of protein synthesis, transcription. So this is going to take place in the nucleus, and we're going to have our DNA molecule in our nucleus. And what's going to happen is we're going to have an enzyme that comes by, and it's going to start to separate our DNA strand. Now it does this. How does it separate the molecule? It breaks the hydrogen bond. So remember that these two DNA strands, I'm not going to draw them on my desk. I don't have paper clips with me right now. But the two strands of DNA are held together by hydrogen bonds. This enzyme is going to come in, break those hydrogen bonds, and it's going to start to separate my DNA molecule. Now, as that's happening, this enzyme is also taking these free RNA nucleotides that are just chilling in the nucleus, and it's going to grab them and it's going to pair them up with the right complementary base pair. So for instance, U, remember there's no T with RNA, it's going to be uracil, U instead, matches with the A, enzyme grabs the the cytosine base pair and matches it up here. And then in this case, we're going to have A match with T, and then G with C. So this enzyme is not only breaking the hydrogen bonds and separating the DNA, but it's also adding our RNA nucleotides. And what I have left now, after it's done doing this, is a connected mRNA molecule. What ends up happening is these DNA molecules, imagine this mRNA is kind of gone further down, the DNA strands are going to zip right back up together, so those hydrogen bonds are going to reform. So I'm just going to bring our DNA molecule out of the screen. So we're only focusing on mRNA. And let me straighten out this molecule. Okay. So we're still in the nucleus at this point, but since I've made an mRNA molecule, unlike DNA, this guy can move out of my nucleus. So my mRNA is going to move into, where is it? Imagine that it's moved out of the nucleus and into the cytoplasm now, right? It can travel like this. Right? The whole reason why it's in the cytoplasm now is because, and this might have been a little bit difficult to kind of figure out how to use these cards, it's going to take it to the ribosome. The ribosome is going to use this message to make a protein. And remember that ribosomes are made out of our RNA. So this is a little extra information right here, but I think it helps show that the purpose of transcription is to make this mRNA molecule. It could go to a ribosome where you could finally make a protein.